This is glycogen molecule during fasting. One of the branched glycogen. There are whole molecules. So just to show, it is one of the branched glycogen part, or one of the part of glycogen molecule. Now you can see here 4 molecules: 1, 2, 3, 4, 5, 6 here. So I told you glycogen. What does glycogen do? Glycogen sits here. Glycogen is very smart. It does not sit here and cut here. So it is not a foolish enzyme. So it will cut here. It will cut here. So 2 glucose 6 glucose 1 phosphate is released. 2 glucose 1 phosphate is released here. 1 1. Now after this 4 tetrasaccharide is, or this oligosaccharide, after this 4 glucose containing oligosaccharide, it cannot come because it cannot cross through. It will be obstructed. This enzyme will be obstructed by this, because this enzyme requires 4 glucose to sit on it. So it cannot cross through this. That is why here comes the next enzyme, second, that enzyme that will transfer this oligosaccharide, trisaccharide. So you can see this trisaccharide will be transferred here by glucan transferase, and this is 1,4 bond. From 1,4 to 1,4 is transferred. Now last bond is alpha 1,6 bond. Alpha 1,6 bond. This bond is actually broken down by debranching enzyme. Here you have seen there is release of glucose 1 phosphate, 2 glucose 1 phosphate. But here release of plain glucose, simple glucose, no glucose 6 phosphate. So this glucose can is permeable and easily cross the membrane without dephosphorylation. Here dephosphorylation, actually phosphatase will come, dephosphorylation will happen, then glucose will go. Here this simple glucose, permeable, non-phosphorylated glucose will be released at this step. So 3-step process, very easy process. The main enzyme is glycogen phosphorylase. I have been telling you about phosphorylase, and then this glucan transferase, and then debranching. Both enzymes are part of same protein. Okay, so you can consider them as one protein catalyzing. This is the one protein. The function of this protein to remove all the branch and put it here. So you can see here glycogen phosphorylase again will act. So this cycle goes on. You can see the bond is also written here. This type of bond is also, and here you can see 1 phosphate. So you can understand yourself also when you study. Now the difference, the main thing is to understand the difference between muscle and liver glycogen. I told you liver is like mother. She is caretaker. She is caretaker. See, liver lives for others. Okay, brain and RBC. Muscle is masculine and selfish. Muscle is selfish. He is very selfish, and you can see that is why muscle will utilize its own glucose. How does it utilize? Then question comes, how does it utilize its own glucose? So I have already actually answered the question. So you can see the steps of glycogen breakdown: glycogen phosphorylase, glucoside transferase, and then debranching. All 3 enzymes will be required to break the glucose to glucose 6 phosphate. Actually, all 3 enzymes is required for glycogenolysis. Now glucose 1 phosphate will convert into glucose 6 phosphate with the same enzyme that you have studied in glycogen synthesis, mutase enzyme. The function of this mutase is to change the position of phosphate group inside the molecule. So this enzyme will reposition this phosphate at 1 position to 6 position. This becomes glucose 6 phosphate. So you can see here also the same thing, the same process happening. But the process, because the process become different here. Why? Because this muscle normally does not have this enzyme. Normally muscle does not have this enzyme, glucose 6 phosphatase. Normally muscle does not have this enzyme. Because of that, this Glucose 6-phosphate, as you know, phosphorylated glucose is not permeable. That is why it cannot come across the membrane, and it will stay inside the liver cell. And because of that, what happens? This glucose 6-phosphate is bound to go into catabolism of glycolysis. So it will go into glycolysis, and that is why this glucose will not be available to blood. And this is how, because of lack of this one enzyme, glucose 6-phosphatase, this tissue becomes selfish. And glucose 6-phosphatase is present in endoplasmic reticulum. That you understand, it is not in cytosolic enzyme, it is endoplasmic reticulum. So sometimes what happens, these are the USMLE, and this does, I have seen the questions, many questions in USMLE. They actually sometimes ask the location of enzyme. So you need to be aware of this. They will ask location of enzyme rather than steps and others. So they will ask things which you don't focus on, and but it is very important. So it is important to know this. So it is endoplasmic reticular enzyme. Now it is not cytoplasmic enzyme. So here it is present. This enzyme is present, normally present, and that is why this glucose will be able to convert into glucose 6-phosphate, will be able to convert into glucose. And this glucose, as you know, this is very permeable, and that is why it will come out of the cell and actually join the bloodstream and reach extrahepatic tissue like brain and RBC and cornea, lens, renal medulla. This is all tissue. It will reach all tissue during which condition? During early fasting condition. Early fasting, glycogenolysis happens during early fasting condition, because we have a limited glycogen storage. If person keeps fasting for a longer duration, so what will happen? Glycogen will be depleted. Okay, and then other process after glycogenolysis, other process will start. What is that process? For up to 1 day, I told you to remember, up to 1 day, 18 hours is appropriate. Up to 1 day, glycogenolysis. Then after 1 day, what will happen? Gluconeogenesis will take over. Gluconeogenesis will happen here also, but the speed of gluconeogenesis, level of gluconeogenesis will be low. Slowly it will start, and after 1 day, gluconeogenesis will take over glycogen, because glycogen storage is depleted. That is why gluconeogenesis is the main important. So both glycogenolysis and gluconeogenesis will supply glucose. Okay, maintain blood glucose. Now see, if any problem happens in glycogenolysis but gluconeogenesis is fine, blood glucose level will not be affected. This is very important. Blood glucose will not be affected. But if gluconeogenesis has problem but glycogenolysis is working well, then there will be not any problem up to 18 hours. But after 18 hours, there will be severe problem. So you have to remember these differences. After 1 day, if glycogenolysis is damaged, so in this 1 day also, this glycogenolysis has problem, this gluconeogenesis takes over early. Okay, but glycogenolysis does not have capacity to go beyond a particular span of time. So imagine things, fasting condition, what happens? Imagine, if you imagine things, you will not have a problem. Imagine yourself being in fasting condition. What will happen? Okay, you are fasting for 1 day. Your glycogen is doing the job. After you have fasted after 1 day, so your gluconeogenesis will do job. And then you can imagine the diseased condition. So you can easily conceptualize the things. So these are the important things. This is one important thing. And what I told you, the maintenance of blood glucose level. So this is important thing. Maintenance of blood glucose level by glycogenolysis, and then gluconeogenesis. Up to 1 day glycogenolysis, after 1 day gluconeogenesis. After 1 day to 1 week, 1 day to actually 1 week, gluconeogenesis keeps going. So after 1 day, gluconeogenesis keeps going. Because RBC cannot survive. Brain will later adapt on glucose, but RBC cannot survive without glucose. Brain will later adapt on ketone bodies. That is derived from fat. Ketone body is derived from fat. This is phosphorylase. You can see use phosphate, molecular phosphate. And why? See, this is a very important concept. When you use water, you call it hydrolysis. Hydrolysis. When you use phosphate, so you call it phosphorylysis. 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 There is one more phosphorylysis we have already studied. Do you remember one phosphorylysis? In nucleotide degradation. Do you remember? Glycosidic bond and that exon and glycosidic bond of nucleoside. Do you remember? PNP, purine nucleoside phosphorylase. So this is also, this enzyme, go and watch that. PNP, purine nucleoside phosphorylase. This enzyme, this phosphorylase does the same thing. It breaks this bond between sugar here and then base and glycosidic bond. So you can go and watch that. You can go and see that in your book and anywhere. So this bond is also broken by different type of phosphorylase, PNP. Here, this is different phosphorylase. So this phosphorylase means hydrolysis means you break the bond with H2O. When you break the bond with using this phosphate group, this is called phosphorylysis. And the enzyme that actually breaks the bond using water are called hydrolase. Hydrolase. Here, the enzyme that use phosphate to break the bond is called phosphorylase. Okay, does it should be, you must correlate it with this. Okay, and this is transferase because phosphate group is transferred to it, and that phosphate group will stay with the glucose. After addition of phosphate, there will be release of glucose 1-phosphate. Here 1 bond, 1 carbon, this is 4 carbon. So this glucose will go and attach to this phosphate, will attach to 1 carbon. That is where glucose 1-phosphate. So that is where phosphate is transferred, and this is called phosphorylase. Phosphorylase similar to hydrolase. Now this is the enzyme glycogen phosphorylase. Glycogen phosphorylase is dimer protein, and it has got at active site, it has got this pyridalxal phosphate. It is very, very important. PLP. So I told you pyridalxal phosphate, mnemonics. If you can remember or you have forgotten, he is, you can keep a very good and man. So what is he for? Heme synthesis, the first step of heme synthesis. Ala synthase. Don't forget this. Remember, I told you this mnemonics during transamination reaction. V for vitamin synthesis. Which vitamin synthesis? Vitamin B3 synthesis. From tryptophan. from tryptophan. Amino acid conversion to vitamin B3 with the help of this vitamin B6. G for this glycogen's breakdown. This phosphorylase. Then N for myelin. Yes, myelin we have already done in MD1, myelin. First step of myelin synthesis. That is transfer of acyl group to actually serine. Acyl group to serine. So serine acyl transferase. Myelin. And then after myelin, you can see A for amino acid catabolism. We have seen amino acid catabolism, transamination especially. Transamination. Other amino acid catabolism also require this PLP, but don't forget this transamination. And N for neurotransmitter synthesis. We will talk all of this step by step. I have given you mnemonic so that you will not lose track, so that I can revise it many times. Neurotransmitter synthesis. So remember 6. B6 has 6 functions. 1, 2, 3, 4, 5, 6. So that you will not confuse this is for part of mnemonics. 1, 2, 3, 4, 5, 6. So B6 has 6 functions. And this is the one of the function. Okay. So glycogen phosphorylase has got B6 at the center. And this enzyme helps catalysis of glycogen breakdown. This is a very, very important concept. Wherever vitamins comes, it becomes super important. So you have to understand this. Now this B6 will catalyze. If you understand B6 catalyze, that's okay. So this is catalytic site present here. Here one and here one. So you can see, now you can solve this question. Which of the following enzyme is responsible for glycogen breakdown? A is the enzyme, glycogen phosphorylase. And what you knew about the glycogen phosphorylase. Glycogen phosphorylase through that cartoon. So don't forget that also. This is the same thing. The same enzyme I have been showing you in the cartoon, the same enzyme I have been showing in the cartoon. Okay, and correlate this enzyme with this, because these are two pictures. You will actually forget this. You will consider this as different, this as different. So this man actually is the same. This actually body is this, like this. Okay, so understand this. So this dual structure is actually cutting this, chopping, and then it will go and it becomes stagnant at a position. It cannot cross that 4. It cannot cut last 4. It cannot chop last 4 glucose. So liver glycogen contributes to maintenance of glucose, but not muscle glycogen. You already know. Which of the following enzyme is absent in muscle? Yes, there is not a big problem. You all know which enzyme is absent in muscle. C, normally absent in muscle. Now, which of the following is responsible for hydrolysis of this alpha-1,6 glycosidic bond present at the branching of glycogen molecules?
D.
Yes, D is the answer. So you know this. Now, glycogen phosphorylase is responsible for breakdown of glycogen 2. So glucose 6-phosphate is not produced. Glucose 1-phosphate is produced, and glucose 1-phosphate will convert into glucose 6-phosphate by mutase enzyme. And you add the substrate, you will get the mutase, phosphoglucomutase, mutase enzyme.